Blackleg is perhaps the most important disease of canola in Canada and worldwide. While it has not caused extensive yield loss in Canada in recent years, it was the most damaging canola disease through the 1980s and early 1990s. This disease has the potential to cause significant yield loss again if it is not managed carefully. Blackleg is caused by a fungus called Leptosphaeria maculans. The blackleg disease is initiated from old canola residue that was infected in the previous canola crop. The blackleg fungus in this residue produces fruiting structures called pycnidia and pseudothecia in early spring when it warms and whenever it becomes wet. Pycnidia appear first usually on fresh residue from the previous year. Pseudothecia appear later, since these structures are the result of sexual mating between fungal strains, thus appear about 9 months later in Canada. Pseudothecia is more common in older canola residue. Both of these fruiting structures produce spores, pycnidiospores in the case of pycnidia, and ascospores in the case of the pseudothecia. Pycnidiospores do not travel very far, usually just a few meters. While ascospores can travel tens and hundreds of meters. These spores germinate on the surface of a canola leaf and seek out either a stomata or a physical wound in which to infect the leaf. Once inside the leaf, it kills the surrounding cells and produces a lesion. These lesions will typically form new pycnidia and release pycnidiospores to infect more leaves. Some forms of resistance in canola operate at this point of infection by killing the cells around this point of infection prematurely, which stops the fungus from advancing further. This type of resistance is often called major gene resistance or seedling resistance. If the plant does not have resistance, then the fungus produces hyphae that travel through the vascular tissue to the basal part of the stem. For reasons not yet known, it is at this point that the fungus leaves the vascular tissue, causes lesions, and necrotizes the surrounding tissue. This produces the cankers we often associate with blackleg, which cause yield loss by preventing water and nutrients reaching pods and developing seeds. At this stage is the best way to ID blackleg. It produces a black pigment in the interior of the stem, and clipping a stem open will reveal if the disease is present. The second form of blackleg resistance is called minor gene resistance, or sometimes called quantitative resistance or adult plant resistance. This form appears to operate by slowing down or preventing the formation of stem cankers. If plants have neither form of resistance, then the yield loss can be severe if conditions are right for the development of blackleg. Blackleg can be controlled by preventing infection from occurring. Long crop rotations were the primary method of managing this disease prior to blackleg resistance. Rotations that take a break from canola for 2 or 3 years is normally enough time for the majority of canola residue to completely decompose. Once the residue is gone, most of the disease risk has been eliminated. But if a canola grower notices blackleg levels increasing in a field of resistant canola, then a lengthening of the crop rotation is needed. Other mechanisms of removing canola residue from a field have not worked very well. Conventional tillage does not remove enough residue from a field to make a difference. Burning residue does not burn deep enough to remove the basal stem parts where the majority of the fungus resides. But the best tool for combating this disease is resistance. Blackleg resistance, introduced in the mid-1990s, has dramatically reduced the yield loss to this disease and reduced concerns as well. But blackleg resistance is not infallible. Canola growing in tight rotations does put pressure on Leptosphaeria maculans to overcome that resistance. The fungus Leptosphaeria maculans is not a uniform entity. It is composed of many races in the field. Some of these races are capable of overcoming some of the resistance genes that we have in canola. When a resistant variety is grown in the field, those Leptosphaeria maculans individuals who possess the genes that allow them to infect the resistant variety will become more predominant. This is why growing the same resistance in a field in a tight rotation will eventually lead to that resistance failing and the canola crop becoming susceptible. Blackleg needs to be managed correctly in order to prevent it from becoming a major disease in canola again. The potential is there that it can. When blackleg becomes a problem in a field, it not only affects the yield of the crop, but can also affect whether that crop can be marketed. Blackleg can become a trade issue for importing countries that do not have Leptosphaeria maculans in their domestic crop. Canola growers need to ensure that they do all they can to keep blackleg to a minimum for their own profitability and to prevent the loss of markets. Attention to scouting for this disease, rotating crops, and rotating blackleg resistance is essential for the success of this crop.